My name is Maria Walliser. I'm a member of the Adult School Board and co-chair the Lectures Committee with Marcia Snowden. My role this evening, honored though I am to have it, is a pretty superfluous one. If there's anybody in the hall who doesn't know who our speaker is, meet me outside afterwards. I'll clue you in. But tonight I want to tell you about some of the roles that President Tillman has played that I find, it's personal, most significant. In June, as most of us know, Professor Tillman became the 19th president of Princeton University, the first woman, the first scientist, but not the first Canadian. Since then, she's worked in many ways that continue to change both the face and the culture of Princeton University. And she has, over the course of her life and career, successfully filled many roles. First, she's the mother of 2 children. That's enough right there. Second, she's been a beloved and superb teacher. From teaching high school in Sierra Leone, West Africa, after earning her honors bachelor degree at Queen's University in Ontario, and as a professor at Penn, at Temple, and then in her own lab here at Princeton. I personally have heard from her graduate students what a superb teacher and supportive mentor she was to them. One student, and I have to qualify this, I found this story in an old print, so I can't, vouch for its reliability. But nonetheless, one student supposedly carved her initials in the back of his head after the semester was over to celebrate her teaching. So it's no surprise that she was recognized with the President's Award for Distinguished Teaching in 1996, before she was president. She is, of course, as we all know, a world-class molecular biologist. Having earned her PhD from Temple, she was part of the team that cloned the first mammalian gene, and then continued making breakthroughs as an independent investigator at the Institute for Cancer Research in Philadelphia, and after coming to Princeton in 1986 as an investigator for the Howard Hughes Medical Institute. She has a slew of honorary degrees. I counted 25, and has won many awards, more than that. Among them, in 2003, the Lifetime Achievement Award from the Society for Developmental Biology, and in 2007, the Genetic Society of America Medal for outstanding contributions to the field. Another Tillman role is that of builder. In 1998, she became the founding director of the university's Louis Sigler Institute for Integrative Genomics, and a founding member of NIH's National Advisory Council of the Human Genome Project. In her presidency here, we have seen the creation of the Lewis Center for the Arts, the Lewis Science Library, the new chemistry building, fondly known as the Taj Mahal on campus, the Andlinger Center for Energy and the Environment being constructed on the corner of Prospect and Olden, and the Neuroscience Institute rising daily on Washington Road. As a gatekeeper, no, forget that, gate opener, and a consensus builder, her leadership has changed the face and the faces of this campus through diversity initiatives, increasing the size of the student body, and opening Whitman College as the 6th residential college, and offering students the option of a 4-year residential college at Princeton. There are now 2 more of those. She is famous nationally for her support of young women in science, and her support and appointment of women in leadership positions has led to a notable increase in the feminine faces of leaders across the country. As an example, look at this morning's New York Times, an article about Maria Chloe, who President Tillman appointed as the first woman to be Dean of Engineering here at Princeton, and who is now president at Harvey Mudd College in Claremont, California, increasing geometrically the number of women in computer science there. Then there are her contributions to the community. She created the President's Lecture Series, which is free and open to the public. She supported the inauguration and continuance of the Princeton University Prep Program, which helps low-income, high-achieving high school students from local communities between Princeton and Trenton prepare for college. And they do. About 95% of them have gotten into good four-year colleges. She also helped us create this series tonight, and we are very grateful to her for that. Extremely important to me personally, and maybe to some of you, she's an avid basketball fan. I often see her at the games. I often see her at student concerts and other events as well. There's lots more, of course, but rather than use up any more of your time or hers, I'd like to bring her into tonight's role, which is as our lecturer on her own special topic, genomic imprinting, a genetic arms race. President Tillman.
Thank you for that beautiful introduction. I only wish my mother had been here. She would have loved it. It is wonderful to be able to participate in this lecture series on science. I've heard rave reports from a number of you who participated in the lectures that have preceded this one. Some of our most luminous science faculty have been lecturing in this series. So I feel as though I have a very serious challenge ahead, which is to do justice to the other faculty who've talked in this series. It is, I also want to say, just a real pleasure to have a chance to speak to my neighbors. I've been a resident here in Princeton for 26 years, so every one of you is my neighbor, and many of you I know have been for many, many years. So this is really a wonderful opportunity for me as well. Finally, it's a wonderful chance to talk about science. I often am up on my feet having to talk, but very rarely anymore am I given the opportunity to talk about my first love, my first professional love, which is molecular biology, genetics, developmental biology. So you have given me a gift in giving me this chance to come and speak this evening. And what I want to talk about is a very unusual genetic phenomenon that I began studying in the early 1990s and really continued to study right up until the time that I was booted up the hill. And that is called genomic imprinting. And it is one of many examples of something that is now one of the hottest areas in genetics, which is called epigenetics. So I thought I'd begin by giving you just a sense of what epigenetics is. I teach a freshman seminar every fall, which I call How the Tabby Cat Got Its Stripes. Because one of the things you learn when you teach at Princeton is the title is everything, getting students to sign up for the class. And it gets me lots of applications. But it's really, this course is really about what I tell the students is the wild and wacky world of epigenetics. And these are just 4 epigenetic phenomena that you can actually see, without a microscope. Here's the famous tabby cat, whose pigmentation you can see varies from various regions of the body. And that's an epigenetic phenomenon. I'm going to explain why. Wonderfully variegated petunias are in fact variegated corn. Here is maize, which is a very famous genetic organism. The variegation, the differences in color that you see here is a consequence of an epigenetic, not a genetic, an epigenetic phenomenon, and I'll explain that. And one of the most unusual epigenetic phenomena, and one that I have convinced one of my seniors to write a senior thesis about next year, is mad cow disease. So this is a cow that is suffering from a disease called a prion disease, bovine and spongiform encephalitis, and this disease is an epigenetic disease. So there are many examples of epigenetics in nature, but what does it really mean? So I thought I would start off by saying what it isn't, which is standard Mendelian genetics. You know, that for those of you who took biology probably at any time learned in the textbooks his famous experiments with peas, where he began with a pea that was green in pigmentation and one that was yellow in pigmentation. And when he crossed those, and of course he didn't know about genes at this point, he only knew that plants had different colorations. What one got in that first generation were only green peas. Now we know why that is so. We know that what he was following in his cross was a gene that is responsible for the green pigment color. And in the green plant, there were 2 copies of this gene, because this is a diploid organism, 2 copies of producing a protein that produces the green pigment. And in the yellow plant, that gene, that very gene, had a mutation in it. And the consequence of the mutation is the gene didn't function, so no pigment protein was made, so the plant was not pigmented, did not have the green chlorophyll color, and so the plant was yellow. And when you cross them, every one of these plants was going to get one of those chromosomes from one parent, one of the chromosomes from the other parent. So all of these first generation plants had a green, fully functional pigment gene, an inactive gene, but because this one copy was enough to produce pigment, all of the plants were green. Okay? Classic Mendelian genetics. And we refer to the green version of this gene as dominant because it is the one producing inactive protein. Now, what happens in the next generation when Mendel took one of these plants, these first generation plants, and crossed them with each other? Well, again, on the assumption that you're going to get one copy from one parent, one copy from the other parent. If you just do the math, 25% of the offspring are going to get the green from, let's talk, from this parent, a green from this parent, fully green, and the plant's green. About half of the plant, in fact exactly half if you do enough of them, are going to get a green copy from one parent and the yellow copy from the other. And it could happen in this direction or it could happen in the other direction. And these plants, like this plant, are green because each one has an active pigment producing gene. But a quarter of the time, you're going to get the mutant copy from this parent and the mutant copy from this parent, and 25% of the offspring are going to be unpigmented. They're going to be yellow. And this is what we all learned in the textbooks. So, what happens when there's an epigenetic phenomenon occurring in a plant? So in this case, the phenomenon I'm showing you happens in not peas, but in maize, in corn, wild corn. And you begin with a plant that is actually relatively unpigmented. It's green because the pigment color in maize is purple. So you assume that this is a plant where the pigment genes are not working well in some way or another. And you cross it to a fully pigmented plant. And these are dark, dark purple. Really very, very pigmented. And what you get in the first generation is not pigment. What you get in the first generation is the lack of pigment. So already that's a bit of a surprise, right? You'd expect that these were going to be pigmented. In fact, they're green. But it gets even weirder. Because if you do a cross now where you take one of these plants that is now green and you cross it back to the purple plant, expecting we're going to get some purple plants back because 75% in this cross are going to be fully purple, like this parent that we started off with. You get 75% inheriting the purple genes that originated from this purple plant, but 100% of the plants are green, not pigmented. So something has happened to this pigment gene in this first cross that basically silenced it in some way. It's not a mutation. The DNA is exactly the same as it was in this purple plant. But when you combine this gene and this gene, somehow or other, this gene gets converted from an active gene to a silent gene. That's epigenetics. And I can tell you that I used to go to epigenetics meetings in the 90s when a very fine corn geneticist named Vicki Chandler used to stand up and talk about this phenomenon. And we'd all go, voodoo, voodoo. This is inexplicable. This is magic. We're never going to be able to figure this out. And we really didn't figure it out until the last 10 years. So, but we now at least have a pretty good idea of what happened in those plants. And so this brings me to the definition of epigenetics, which is the study of heritable changes. That change in that pigment gene was heritable from generation to generation, the silencing of that gene. And actually, Vicki had crossed those plants for 25, 30, 40 generations and could never revive that silent gene that was no longer working. So it's a heritable change in gene expression or cellular phenotype, that just means the way the thing looks, caused by mechanisms other than changes in the underlying DNA sequence. So how can this happen? So we have to begin with how the DNA, which is of course the material out of which genes are made, how DNA actually lives inside a cell. And so way down here at the very bottom of this slide is sort of a cartoon of the famous double helix that was discovered by Watson and Crick back in 1953, composed of just 4 bases or nucleotides, A, G, C, and T, organized in these anti-parallel double helical structures. That does not exist in any cell, in any organism, that's known in nature. Instead, DNA is always associated with proteins. And in eukaryotes, which are multicellular organisms like us, that DNA gets wrapped around proteins. And these are the proteins which are called histones. There are 8 of them in each one of these little cores. And the DNA, you can see, just gets wrapped and wrapped with about a periodicity of about 140 to 180, 190 bases. But that's not the end of it. It's not just one run. Then what happens is once you have these beads on a string, which is how we refer to them, they get further kind of compacted. And the way I always think about this is think about a ball of string. You know, you start off with the string being really long, but you know, as you kind of wind and wind and wind, you can compact that string more and more and more. And so you get what is called a 30 nanometer chromatin fiber. But that then gets organized further in the nucleus. It gets organized further until finally you have this highly, highly compacted structure, which is a chromosome. So you can't think of DNA as kind of naked. You have to think of it as totally compacted around these proteins. And that level of compaction can vary from gene to gene. So for example, you could have relatively little compaction. There's actually, you know, space between those various histone octamers. And when a gene is in that relatively open state inside the nucleus, the gene is capable of being expressed. So this would be sort of what a gene looks like when it is being expressed inside of the cell, like the pigment genes we just talked about. But that same gene can also exist in one of these highly coiled, highly condensed structures that look something like this 30 nanometer fiber. And it won't surprise you that that is the structure that a gene adopts when it is silent, when it is not being expressed, when no copies of it are being made inside the nucleus, mainly because the copying machinery cannot get to the gene. The gene is just tied up in a sort of a ball of protein. And so genes can exist in this state, or they can exist in that state. The important thing is the DNA underneath both of those is identical. So how do you get a gene either in one state or in the other state? That's epigenetics. Now there's another modification that can happen to a gene that will affect whether the gene is on or off. And that is one of the 4 bases, cytosine, is capable of having a methyl group, carbon and 3 hydrogen atoms added to this carbon position in this 6 carbon ring, or 4 carbon 2 nitrogen ring here. And so you could have a gene where these cytosines are unmethylated, and when, in general, it's not universally true, almost nothing in biology is universally true. When these cytosines are unmethylated, the gene tends to be on. But when these methyl groups, particularly the ones around the beginning of a gene, are methylated, that tends to lead to the gene being off. So there's 2 ways of sort of manipulating a gene inside a cell, either on or off. Okay, now, we're ready now to talk about genomic imprinting and how it is a very unusual epigenetic phenomenon. So, to introduce imprinting, I have to go back to early mammalian development, or the development of any multicellular organism that reproduces bisexually. And of course, all of this is familiar to you. You begin with an egg and a sperm, and the dots inside each represent the what we call the pronucleus, which are the chromosomes that are being inherited from the egg and from the sperm. 23 single chromosomes from the egg, 23 single chromosomes from the sperm, and when they come together, as they do once the egg is fertilized, and these pronuclei actually fuse with one another, what you now have is a fertilized egg poised to begin development, where half of the genetic contribution is from the mother and half is from the father. And off you go. And you can see in this picture of mouse, this is a fertilized mouse egg, and this is the male pronucleus, and here's the female pronucleus. The reason I can tell is because the male pronucleus tends to be just slightly bigger than the female pronucleus. You can actually tell them apart. They're about to fuse, and once they fuse, cell division begins, and you can see that this 1-cell embryo is about to become a 2-cell embryo. It's almost finished cell division. When these 2 separate from one another, you will now have 2 cells, and you'll go on to 4 cells, and so on. And that's how normal development works. Now, in the 1980s, a very old friend of mine, who I collaborated with for years, asked a really straightforward question. And the question, he said, is, okay, we know that each contributes half the genetic information. Do you have to have 1 from mother and 1 from father? Or could you have 2 from father or 2 from mother? And so Davor Solter, who is the person I'm referring to, did this incredibly difficult, at the time, experiment using nuclear transplantation. So he took a fertilized zygote, which is just a word for a fertilized egg, and he went in with a pipette, a very fine pipette, and he pulled out that large male pronucleus, and then he went to another fertilized egg and he replaced the male pronucleus with a female pronucleus. So his argument was, gee, now that egg has just as many chromosomes as this egg. Can it go on to development? And the answer was absolutely not. 100% of these, what are called gynogenotes, because they only have female genetic information, failed to develop. So then he did the reciprocal experiment, and that is he went and removed the female pronucleus from a normally fertilized egg, replaced it with a male pronucleus, and said, well, what about if you have two fathers? What if all the genetic information is coming from the father? Does that work? And again, the answer was no. It didn't work. And so Dauber had a lot of hypotheses about why it is that these two embryos won't develop and this one will. But the answer turned out to be a little surprising, unprecedented as far as we knew in nature, which was that genes are active on or silent depending upon which parent you inherit them from. And these were called imprinted genes because what your parent is doing is imprinting a gene on by determining whether it is going to be on or off in the offspring. So we've already been talking about normal Mendelian genes. Here's the mother's copy, here's the father copy. Normally both are on. That's how 99% of all genes work. That's why Mendel's experiments worked. Imprinted genes look like this. Here's the mother's copy. It's inherited in an active state. It's on, it's working, it's doing exactly what it should. But the father's gene, the gene that you inherit from your father, is covered with these condensing proteins, these histones. And furthermore, it's often methylated all across the gene. And as a consequence, this gene is off. So it really disobeys Mendel's rules about how genes must work. So a big question when we began working on this problem. And I began working on it by really stumbling on an imprinted gene, one of the very, very first to be identified. And so the first question we said, well, how can this be? These genes are swimming in the same nuclei, you know, with the same kind of regulatory apparatus. How can one be on and the other one be off when they're in the identical sequence? And so the answer turned out to be that they are organized this way at the only time when these chromosomes are apart, which is during the formation of eggs and the formation of sperm. So here's what happens. You start off with primordial germ cells in early embryos. These are the cells that will become the eggs or the sperm, depending upon whether the embryo is female or male. And when the oocyte begins to package its DNA into these chromosomes, it packages these imprinted genes in a way that is different in the oocyte than it is in the sperm. And so when these two come together in the zygote for the very first time they're ever together, they're already different. And what is really remarkable about imprinted genes is that difference is sustained for the rest of the life of the offspring. So this is real parental imprinting, if you want to think about it that way. Now, this posed a huge paradox for biologists interested in genetics, because imprinting results in offspring that only have a single active copy of what in some cases are very critical genes. And all of what we knew about evolutionary biology would have suggested that evolution would have selected against this phenomenon over and over and over again. There is a huge benefit, which you saw in the Ps, of having two active copies of a gene, which is you can suffer a mutation in one and still be absolutely fine. In fact, people hate it when I say this, but every single person in this audience has, on average, 24 lethal mutations in your genome. The reason you're sitting in this audience is because those lethal mutations are in one copy of an essential gene, that the other copy is working perfectly well, as we can see. So how did this imprinting business come into being? And just to show you that it really does have an impact on human physiology and pathology, there are a number of diseases, human genetic diseases, that are associated with imprinted genes. This is one of them, called Beckwith-Wiedemann syndrome. These are identical twins. These are genetically identical twins. This one is suffering from Beckwith-Wiedemann syndrome. This is the unaffected one because of a misregulation in a single imprinted gene that happened early in development in this twin and did not happen in the other twin. And there are a whole series of sequelae that result from this. The most serious are the development of embryonic tumors like retinoblastoma, hepatocellular carcinomas. So it's a pretty serious disease. These are even more serious diseases. This is a child with a disease called Angelman syndrome. This is a child with a disease called Prader-Willi syndrome. These children have loss of function mutations in their one active copy. And because the other one is silent, it can't compensate. And as a consequence, these are very serious diseases of children, both of which result in mental retardation. So these imprinted genes are important genes. That's another way of saying this. So it becomes even more surprising that evolution didn't select against this phenomenon. So the question we got really interested in, I'm a molecular biologist, but evolution is one of the most fascinating things that biology studies, is why? What is the purpose of this imprinting? How did it arise in evolution? So we started looking for clues. And the first clue came when we began to analyze mutations in imprinted genes in animals, obviously. And one of the things that we discovered is that more often than not 100% of the time, again, biology never cooperates that way, but more often than not, when you made a mutation in an imprinted gene and therefore had no active copy, what was affected was fetal growth. And these little runts have mutations in one imprinted gene that clearly is required to promote growth. The offspring are small, and these on the end have mutations in a gene that slows down growth. And when you mutate it, the animals get oversized. So somehow or other, fetal growth is part of the story. And the second clue came when we began to look at where does imprinting happen in the animal kingdom? What animals imprint, what animals don't? And the answer there was pretty clear, and that is, it is only found in mammals that have placentae. So it happens in humans, rodents, cows, pigs, horses, wallabies, and opossums. Because, of course, early in development, those marsupials do begin gestation inside the mother with a placenta. The difference, of course, is that they are delivered about halfway through gestation, crawl up the mother's stomach and into the pouch, and spend the second half of gestation feeding with milk as opposed to through the placenta. But we looked very carefully in animals like chickens, and we could find no evidence for imprinting in chickens, nor in the fascinating platypus, which is a monotreme. It's a mammal, but it's a mammal that lays eggs. So it lays eggs, but the offspring are fed through lactation. So it's a hybrid mammal, if you want to think about it that way. But other investigators checked the platypus. We tried so hard to get platypus samples. For years, we pleaded with Australians. But it turns out there are laws about what you could do with platypi. And we didn't apply. So there's something about the placenta. So let's just review very quickly why the placenta might be really important here. So this is just to remind you that here is the female ovary that produces eggs that are fertilized right here in the fallopian tube. The fertilized egg actually begins development as it goes down the fallopian tubes, enters the uterus, and then does a truly amazing thing, which is this embryo, this 1632 cell embryo, invades the wall of the mother's uterus. It's an invasive species. And all of you who've had teenagers will understand what I'm talking about here. It really invades, and it sets up this remarkable feeding mechanism. So here is a mouse embryo. Here is the umbilical cord. Here is the placenta. And it's through this placenta that nutrients are coming from the mother into the embryo. And there is a lot of chemical communication that is happening through all of gestation between the mother and the embryo. It's a truly quite remarkable thing. So, next clue is that when we began to look at how those imprinted genes affected growth, thinking about what I've just told you, there was an absolute rule. This is one where it really applied 100% of the time. And that is the mother was silencing genes that promote growth. So they were, through this silencing mechanism in the egg, was kind of slowing down the growth of her offspring. And the father was silencing genes that normally reduce growth. In other words, what he was trying to do is to promote the growth of his offspring. So you had parents with quite different intentions here through this genetic mechanism. And that turned out to be a very important clue. And it led a wonderful young man named David Haig, who I also had a wonderful collaboration with over the years, who's at Harvard. He's actually a much happier person if he looks in that picture. It was much more fun collaborating with him than that might imply. Developed what I think now the entire field believes is the correct explanation for this genetic phenomenon. And it's a wowzy. So here's what David said. That in polygamous species, and polygamous ends up being important, in polygamous species, and that means that females are going to mate with multiple males and vice versa. Humans are polygamous and have this imprinting. But this applies to polygamous species. The paternal genomes, so the genomes coming from fathers, that will be selected in a population are those that increase the likelihood that his offspring will be able to garner maternal nutrients during gestation. Remember, the mother is the only one providing resources to the offspring during gestation, even if it's at the expense of the mother and her future offspring. Because his goal, evolutionarily, is to send as many of his genes into the next generation as possible. So he's going to silence any gene in his genome that's going to slow down the growth of his offspring. Now, why is polygamy so important in this? because of this last part of David's idea, which is that if you have a monogamous couple, then the parents have equal interest, not just in the current offspring, but in all future offspring. But in this case, this father knows that his next set of offspring are going to be sired by a different mother. And so he has no investment at all in her future fecundity. Now, what about maternal genomes? Well, maternal genomes that will be selected in such a population are those that favor equal allocation of maternal resources among all offspring, including future offspring, irrespective of paternity. So she also is trying to maximize the transmission of her genes into the next generation. And the only way she can do that is to counter what the paternal genome is trying to do, which is to slow down growth so that her offspring, so that she is going to be capable of having future offspring. And one way to really imagine how this may play out in evolution, how it might start it, is to understand that it probably began with very ancestral rodents. Mice, rats, animals, very small animals. And one of the things we know about those animals is that in the wild, it is very common to have multiple paternity within a litter. That means that females are mating with multiple males in one 24-hour cycle. So now picture having paternal offspring in the same uterus, fighting for the same capacity to invade the uterine wall, having the same competition for those maternal resources. You can imagine, in that case, you're going to set up a situation where you're going to set paternal genomes in competition with one another. And which is the genome that's going to win? The genome that's going to win is the one that can extract the greatest amount of resources from that mother in that litter. And that's probably how this whole thing really got started in the first place. So how could you, I've often tried to figure out how to actually represent this visually, because I tend to be a visual thinker. And this is the best I've ever been able to do. So here we have sort of, in early evolutionary time, we have a little mouse. And the little mouse is probably sort of an optimal size for the survival of the species. And we have sort of an equal competition between the maternal and the paternal genomes, which is what is represented here, leading to this very optimal growth rate. So what happens? Well, you get variation in the population. That's one of the great things we learned from Darwin, is that evolution works when there is variation in the population. And a female mates with a male who has figured out a way to use epigenetics to silence a growth-reducing gene. And so you now get an unequal, basically an unequal battle between the maternal and the paternal genome, with the paternal winning. And what you get are bigger mice. Now you can say, well, why is that so bad? Isn't that what the male has been trying to accomplish? The answer is yes for the individual. But for the species, one of the things that we did when we generated these oversized mice is we actually measured over many generations the fecundity of the mothers producing those oversized mice. And it will not surprise any woman in this audience to know that when a mother was delivering, generation after generation, these oversized offspring, she had many fewer offsprings overall in her lifetime. So that becomes disadvantageous to the species. So you get this battle, and so what happens? The maternal genome figures out a way to counter what the male genome has done by silencing a different gene, by silencing one of those growth-promoting genes. And now what you get is you get sort of an even balance. You go back to a more optimal size until, once again, there's variation. A paternal genome is selected, gives you a short-term advantage. That short-term advantage is then countered by a maternal genome that arises in the population in exactly the same way. And you get, once again, back to a reasonably optimal size for the species. And so it works exactly like an arms race. You know, you start, right? You start with everybody having the same number of nuclear weapons. Somebody breaks the rules. You've got a counter. Somebody breaks the rules. You've got a counter. It is an arms race. And it functions exactly like a genetics arms race. So, one of the wonderful things about David's hypothesis is it made a number of predictions. It predicted, for example, that imprinted genes will be involved in fetal growth control, and I've told you that that's true. That paternally expressed genes will promote growth, and I've told you that's true. Maternally expressed genes will retard growth. Imprinting will not be observed in oviparous, that means egg-laying animals. And perhaps the most intriguing prediction was one that imprinting will be absent from a monogamous species. Well, as you can imagine, this became sort of the holy grail. Could we find a monogamous mammal? We'd ruled humans out, and but we thought we came about as close as we could find. And it's a little mouse called Paramyscus polyanotus. And Paramyscus polyanotus lives in Florida. Go figure. It has developed a monogamous lifestyle primarily because it is living under conditions of very poor nutrition. It's living on the beaches, basically, in Florida, which for people is great, for mice is not so great. And as a consequence, when resources are so limited, there is a real advantage to a species to pair bond. And we got very interested in this mouse and a way to compare it to a very similar mouse. These are different species, but they're related in evolution, called Paramyscus maniculatus, which was clearly a polygamous animal. So I had a wonderful evolutionary biologist named Paul Verana who came to my lab to study these animals. And we spent a lot of time studying imprinting in them. And one of the things that he definitely showed is that Polyanotus does have imprinting. So the one prediction of the Higgs hypothesis that we couldn't verify was the prediction that a monogamous animal would not have imprinting. That led to huge battle among Paramyscus specialists who said, well, they haven't been monogamous long enough, you know, it's not enough evolutionary time, and they're not really monogamous, you know, if one dies, the other will mate with another, and that's not true pair bonding. But the truth of the matter is that that was one of the predictions that we could not verify. Nevertheless, in studying these animals, one of the amazing things that Paul was able to show is that if you cross a Polyanotus to a maniculatus, all imprinting gets screwed up in these animals. They can't read each other's imprints. And I'm going to skip way ahead to the conclusions that he reached, and then I want to end with some real implications about imprinting. So one of the things Paul was able to show is that imprints are evolving very rapidly in mammals. And that's not surprising, given what I've told you about the arms race. It's still happening. It's still going on. There are genes that are imprinted in us that are not imprinted in mice, and vice versa. So this is a rapidly evolving genetics arms race. differ between species. And the last point I want to make for you is the really important point that imprints are metastable. And that's one of the things Paul showed when he crossed these closely related species. And that has, as it turns out, some really important implications for human reproductive technology. Because when these precious zygotes, these fertilized eggs, are put outside the body or in culture, these epigenetic differences on their chromosomes are not stable in that environment. And so one of the things that we know is that the nuclear transplantation that produces cloned animals like Dolly, here was the first cloned animal, the vast majority of cloned mammals die in utero, and they die grossly oversized. And when you go in and look to see what's wrong with them, what's wrong is they have misregulated their imprinted genes, and as a consequence, genes that should be silent have been activated. So it's made the cloning of any animal a much more difficult thing, but it has also made much more difficult the use of embryonic stem cells, ultimately as therapies for the treatment of disease, because embryonic stem cells are derived from 8-cell embryos when these imprints are still quite metastable. And when you take embryonic stem cells and you put them and you grow them in culture, we now know that the imprinting that's in these cells has gone awry as well, in some fraction of the embryonic stem cells. So I think this is going to be a serious challenge for those who want to use embryonic stem cells ultimately in order to cure very important and debilitating diseases that we would like to be able to cure with stem cell therapy. And then the last image is just to point out that any in vitro fertilization that is done, again, requires taking these fertilized eggs that are in this very sort of fragile, metastable state when it comes to imprinting. And we know that some of the lack of success of in vitro fertilization, is far from a perfect procedure, is caused by these imprints that go awry. So that's where I want to end the lecture by saying that this has been a journey to try and understand a very unusual genetic phenomenon in mammals that's unique to mammals, and one that clearly has some implications for how we think about all of the reproductive technologies that involve putting very early human embryos into culture. And I'll end there, and thank you for your attention.
We'll take a few questions.
Yeah.
So if there's this discrepancy between paternal genes that are promoting growth promotion and paternal genes that are not to, are there examples of females that can recognize males that have this trait and
So I will tell you part of the story that will be a partly serious and partly humorous answer to your question. Over the years, one of the truly extraordinary things that has been discovered about imprinting is that it doesn't just control fetal growth through regulating fetal genes that are expressed during fetal development, but it also regulates the number of oxytocin-producing neurons in the female brain. Now, oxytocin turns out to be a very important hormone that regulates maternal behavior. So when mothers, first see their offspring, and I did it twice, and fall instantly in love and become completely cuckoo about this thing, it's being driven by oxytocin. And And imprinting is controlling the total number of oxytocin-producing neurons in maternal brains. So when my daughter announced that she, was thinking about getting married, I immediately said that I want to measure the amount of oxytocin in her, and intended because of the following experiment. This is such a potent hormone that if you take a male rat, and male rats are terrible fathers. I mean, really, they'll eat their offspring before they take care of them. And you inject a male's brain with oxytocin, that male will start grooming, building nests, behaving exactly like a female. So my advice to all of you who have marriageable daughters is to require an oxytocin measurement before you allow your daughters to get married. It's an amazing phenomenon. Yeah.
In the spirit of your presentation here, is something wider that we learn in terms of warfare of mankind and the social scene. There are all sorts of wild things. Hundreds of people killed each other in the last century.
Yeah. I don't think so. There is nothing in the phenotype that we've seen of animals that have mutations in imprinted genes, which is how we learn about what the function of these genes are, to suggest that aggressiveness of any kind is involved in this. In fact, the oxytocin example is almost the opposite example, where it's really a process that is designed to create nurturing of the young. So no, I don't think there's much we can learn from this, I'm afraid. I wish there were. Yeah.
So the last thing talks about how it's difficult to do this in vitro because of imprinting. I'm wondering if that has anything to do with a mother having some sort of like defense against foreign DNA.
Yeah, there were actually some really interesting ideas that imprinting might be related to protection of the mother. For example, one idea was to protect mothers from the formation of ovarian tumors. It's a long story, but to my knowledge, there is no good rationale for saying that this is one of the reasons for imprinting. I think David Haig has got it exactly right. I think it really is that by the combination of polygamy, multiple paternity within a litter, and this very unusual way of feeding the embryo, which is entirely maternal. If you think of, one of the things I didn't say, but if you think about a chicken laying an egg, you could say, well, wait a minute. All of the nutrients inside that egg are derived from the mother. That is true. But a chicken can't go back and renegotiate how much resources it's going to get from its mother. It gets a prepackaged amount. It's a done deal. But because of the placenta and the umbilical cord and the uterine wall, there is a sort of a minute-by-minute conversation happening between a mother and an embryo at all times during that 9 months of gestation. It's really, they're constantly negotiating how many resources are going to go in what direction and how. And it's those unique combination of things that has created this very unusual genetic phenomenon. Yes, sir.
The Mendelian system was discovered many decades, centuries ago, but the green and purple maize epigenetic
Yeah.
was that only discovered recently? Yeah, because obviously that completely violates Mendel. And if Mendel had studied maize instead of beans and beans,
He would have been terminally confused if he had studied maize. It was a good thing he chose peas. Yeah. Epigenetics, if you go back now sort of with, 2012 eyes, you can go back, 78 years in the biology and the genetic literature and see phenomena that people described but couldn't explain. So there were already, biologists confused by some of this non-Mendelian behavior of genes, but the field really took off in the 90s. I mean, this is really a brand new field. Imprinting was discovered in the 90s. The paramutation, which is the maize, was first described back in the 30s, but no one worked on it because it was so confusing. And it was really this woman, Vicki Chandler, who took it on and figured it out. The petunias, all of you who grow petunias and love those variegated patterns, those are all epigenetic phenomenon where the white parts of the petals have suppressed the gene, have silenced the gene, and all the purple parts or the pink parts of the petals are expressing the pigment gene. And different strains of petunias have different patterns, as you know. If you buy one where the purple is all on the outside, it pretty much stays on the outside during your growing season, although it doesn't have to. It could actually switch, and I hope all of you who grow petunias will now be watching your petunias to see what happens. But it's a relatively new field, which is why it's fun to teach freshmen. Yeah. Yes.
I'm a scientist in disguise, but I'm going way back, to the meiosis, when the sex cells are being formed and the chromatin is being compacted or DNA is methylated. Every single egg will then have the same pattern.
Yes. Yes.
So that is just, and so I guess what I'm curious about is because proteins are genetically controlled and are produced by our genome, how is it?
How do they get different?
Yeah, and how is it not? How is it considered not a genetic phenomenon?
So of course, it's a genetic phenomenon in the sense that it's being regulated by genes. It's epigenetic in that you can get these heritable differences. Remember that the heritable difference, the silencing of the gene that is being built in the egg or the sperm comes together in the fertilized egg, and then every cell that derives from that fertilized egg heritably, as the DNA replicates and replicates and replicates and replicates, that same pattern of active or silent is maintained in every cell for the life of the organism. So it's a highly heritable but non-DNA based difference in how a gene gets expressed. And that's why it's referred to as epigenetic. It's not DNA based, it's what happens to the DNA. Yes?
Is there any research going on or going to be in the future that would lead to the direction of these bad genes, eliminating them, scrubbing them out?
So, an unbelievably interesting experiment was done by a colleague of mine at MIT named Rudy Yanish, which I think it's a tour de force. In a way that would take me too long to explain because it was technically very, very difficult for him to do this experiment. He created a mouse that had erased all imprints. Now remember what I showed you with the nuclear transplantation. If you only erase the female ones, you die. If you only erase the male ones, you die. Rudy made this mouse where both were erased and the mouse was absolutely fine. So it really is a balancing, right? It's this arms race. You can't disrupt the one side of the arms race, but if you eliminate the arms race, actually you can get a normal animal. Amazing experiment.
Will it be applicable to humans someday?
Yeah, not with current technology, no. It would be very difficult to do with current technology. Yeah. Yes.
Metastable, you said it's metastable. So if I get the girl side from, just to be extreme, from 15,000 years ago, and I get the male side from right now,
Yeah.
and I put them together, do I get a living creature, or is the key structure that different?
All right. So that experiment has been done by Paul Verana in mouse, not in humans. I don't want to get arrested here. But this, what you just said, Paul did. Because he asked, what would happen if we took these two species that have now been apart in evolution, roughly the length of time that you just talked about? They're reproductively isolated, is the way we put it. And you cross a monogamous mother with a polygamous father. What you got were these animals that actually died. They were so big they couldn't be delivered by the mother. And so it was a lethal event. If you did the cross in the other direction with the polygamous mother and the monogamous father, you got runts. So what it's saying already, before he then went and sort of sorted out what these differences were all about, it said that in the roughly, I think it was 30,000 years that these animals had been reproductively isolated, they'd already started evolving, different imprints. And so they were totally out of whack.
So there's this usual sort of localization.
Wow, yeah, and so we got really excited by this result because what it suggests, this may be maybe one, you never say the reason, but it may be one of the most important things that determine speciation, right? Because frankly, these die, I've already told you that, so it's a completely lethal event. These in the wild would die. Runts don't survive in the wild. So for all intents and purposes, and he went on to genetically prove that these differences were because they weren't recognizing each other's imprints anymore. So it is driving speciation, and there are crosses that have been done that look exactly like that. If you, I'm going to get this wrong. If you cross a camel and a llama, you get tiny, tiny little animals like this in one direction, and you get death in the other direction. If you cross a lion and a tiger, in one direction you get this, and actually in the other direction it dies. So these are ligers. And the other ones are called camas. And these animals, now they're sterile. You know, it's like a donkey and a horse and a donkey, right, which are sterile. So I think we came to the conclusion that these metastable imprints, which are evolving so fast, are probably contributing to rapid speciation. And one of the things we know is that mammals are speciating much faster than other vertebrates. And this may be the reason. Yeah.
Did I understand, Dr. Coleman, that you said that the embryonic stem cells are not really, in the very near future, going to help with the curing of diseases? That we had all been much more optimistic about.
I'm optimistic in the long term, but I think we're looking at years and years. I think there are many technical challenges that we haven't figured out. And it's going to take a long time to figure them out. This is just one of them. There are others as well. We know a lot more than when I started my career about how to take an embryonic cell and direct it to become a cell that we want it to become, a pancreatic cell or a heart cell or a liver cell. But we don't know how to do it reproducibly, and we don't know how to do it to the exclusion of any other cell type. So I can take a culture of embryonic stem cells and largely get neurons, but I won't get exclusively neurons. And if you're going to be introducing these cells back into patients, you want to be really clear that what you're introducing is what you think you're introducing. Yeah.
One more question?
Yes.
How many genes out of the whole genome are observed to be subject? Are we talking about a very small fraction or a vastly large fraction?
Yeah, I should have mentioned that. So we now think there are, roughly between 20 and 25,000 genes in the mammalian genome, in the human or the rat or the mouse for that matter. We know about 100 imprinted genes now, but an incredibly good scientist at Harvard named Catherine Dulac has recently done a study that I think needs confirming. So just take a little bit with a grain of salt what I'm about to say. But her work is suggesting that in the brain, where you want probably maximal variation in cell type, that there are many more genes that are undergoing imprinting in neurons. So the number may actually go up. But right now we know of about 100, and as I said, many of them are critically important in this early fetal development period in producing maternal behavior, exactly as David Haig might have predicted. Did predict, as a matter of fact. Yeah.
Thank you.